Okay, before we get to compliment, just to make sure, have you guys noticed we're almost done with the semester?
Yeah.
It's coming up fast. Have you noticed on your little sign-in sheet that we're like towards the very end? This is so exciting, isn't it? Okay, so I've had people ask me, how many points are left in here? We have about 160 points, okay? And we're getting down to like, the bare minimum here. As far as our plan goes then, we're going to finish adaptive immunity today. We'll finish innate and then talk about adaptive immunity and finish that. And then we'll move on. We have 2 more main lectures. We have our food and water micro, and then we have pathogen attack. And that should take us through the end of the semester. If we get done a little early, we'll watch one of my favorite videos, which are always fun. I always like to share videos with y'all, so you have to watch a video. As far as the lab goes, we have our 3rd exam today in lab. And then we're going to check our results from last time. The lab today that you're supposed to be doing is called the Discovery Lab. And what it is, it's amazing. It was one that was put out by the American Society of Microbiology. And it's basically a list of all these products and how they use microbes, their enzymes, their pigments, how they've used them in history. They talk about how different antibiotics have been made. And so it's called the Discovery Lab because there's all this information about how we use microbes in ways that we might not even think about. Like they're in laundry detergent. Did you know your microbes are in laundry detergent? They use enzymes in there to help break the dirt off of your clothes. So it's a really neat lab. They talk about how antibiotics actually came from a piece of leather at one point because there was bacteria growing on leather. I mean, fun stuff. So that lab is one that you read and then you look for commonalities between certain organisms. So for example, I'll ask you, what do these 5 products have in common and which one is the outlier? So you read about those products and try to figure out what it is that they all share and which one is it that doesn't share anything. And that's what you're reporting on your answers. That's how you can do the lab. Now the reason I'm telling you all this now is because today we have our lab exam. If you want, you can complete that lab at home because it's just reading and then answering questions. So I know after the lab exams, a lot of times people are tired. So if you want, you can go home and work on it. That's usually what my class does. It's actually set up to be kind of an at-home lab. So I would encourage you to go ahead and think about that. And then, of course, we can compare answers when we come back together on Wednesday. As far as your lab from last time, you set up plates. And today you're going to be doing plate counting. We'll go through the calculations on Wednesday. I've learned in the past that when I try to do calculations after an exam, it doesn't work. Everybody's just like, I don't care at this point. They're tired. They're just like, oh, shoot, we're going to stop talking. So what I recommend, take a picture of your plates, find the one that's countable. What's our countable range? Excellent, 25 to 250. I'll give you a hint, that's usually your 4th plate, between your 3rd and 4th plate, so look at those 2 specifically. But take pictures of all of your plates, and then you can count your colonies, and next time, using your counts, we'll go through the calculations together. Make sense? So today, as soon as you're done with your exam, pull your plates over, you can count them or take a picture of them, and then you're out of here. Because the rest of the lab is going to be done at home. Questions, comments, concerns?
For the ones done at home, is it just the lab? We're just going over the actual lab, just reading over it?
Yes, it's all in your book, and it's just one that you just read about. So, I mean, you have until the end of the semester to finish it. Really. It's not like there's anything that you have to get done in a certain timeline. It's not online. It's all in your book. And it's just reading and answering questions. And it's really neat. Everything's organized alphabetically, but it has like diatomaceous earth. It has things like license, but it has all this fun stuff that you can read through. All right? Okay. So last time we were talking, we met about innate immunity. And what was the difference between innate and adaptive immunity? I told you at the very first. What is innate immunity? Excellent. Some immunity that occurs naturally. Is it specific? No, it's not specific. It's going to act the same whether it's a virus, whether it's a bacteria. It's going to act the exact same. So innate immunity, as we mentioned, are things like skin. They're things like fever. They're things like lacrimal ducts. They're things like chemical enzymes in our tears and in our saliva, pH, all of that stuff. That's all going to protect us, and it doesn't matter what kind of bacterium or virus we are. Another part of our defense system, we talked about last time how fevers help us with our defense system, right? Are fevers good or bad? They're good. As long as they're a low-grade fever, we're okay. What temperature should we get worried or start to get a little concerned?
103.
Yeah, 103. If we get to 103, then I want to do something about it just because I want to make sure I'm not in a positive feedback where I keep going higher. 103 is a good point to intervene. So like I said, I tell my kids, suck it up until they get to that 103, and then I'm kind of like, okay, now I'll give you something, but I want them to have a chance for their body to get rid of whatever they're fighting. Complement goes hand in hand with fever. Complement is another natural system, and the way to think of complement is it basically is like a sequence of dominoes. Have you ever seen dominoes falling, like in a long plane? People put together these intricate things. I always wonder how they have that time. Like you see all those dominoes, like I would never have time to do that. But the idea behind this then is you have a protein circulating in your blood. It comes into contact with what we call an antigen. An antigen is a foreign substance. It can be a bacteria, it can be a virus. And some people it can be pollen. And some people it could be shellfish. Okay? So it comes into something that your body recognizes as foreign. And it bonds to that protein. And it's like clicking the first domino in a long line. What that protein does is it binds to that foreign object and it releases an enzyme. That enzyme binds to another protein. That protein releases an enzyme that binds to another protein. And what happens is I get a sequence of about 30 proteins working together, releasing enzymes, and attaching to this antigen. So when they're done, the little antigen that came in has all of these proteins and enzymes attached to it, which in some antigens can actually destroy them. Just that extra weight can kind of pull them apart. If you picture like a little microbe cell with all these things on it, it's too heavy, it just kind of gets pulled apart. Or what can happen with complement is they can tell T cells, which are our antibodies circulating, hey, this thing's foreign. It's like little flags, they're marking it, and the T cells come in and destroy it. Okay, so that's what's happening with complement. Now, the idea behind complement then, like I said, it's 30 plus proteins working together. And it complements our immunity. That's why it's called that. It'll destroy by bursting the bacteria, so attaching to them to make them too heavy. It causes white blood cells to see that there's now this accumulation. So we have our macrophages come in, our neutrophils come in, and it will help with phagocytosis because it marks it for that T cell. Now, when I first started teaching, and I feel really badly about this, I made my students memorize all the complement proteins. I know, I feel like I should email them all and apologize. 30 plus proteins, what was I thinking? It's like, oh, so evil. And they had to memorize this chart. I gave them this chart empty and they had to fill it in. Isn't that mean? Gosh, I was mean when I first started. Yeah, Aaron.
Is this the one that punctures holes?
It does. It can rip things apart. It can pop holes. It can do any of that stuff. You got it. Do you remember it from physiology?
Yeah.
Yeah, and I'm giving a very simplified version of this. So what happens then is we have an antigen coming in. Now at the top, it shows the different pathways that can trigger our complement. So we can have, for example, a trigger that's occurring by a pathogen. We can have a trigger that occurs by some sort of a chemical or sugar they release. Or we can have a chemical that's on the surface of their cells. So these are all going to trigger complement. Now, once they trigger, the first domino binds to them and releases an enzyme that triggers all the other proteins in a row. So again, they're like little dominoes. So when we're done, all of these proteins and their enzymes are attached to the cell that they're trying to target for destruction, or the antigen, okay? So the proteins then are gonna act like little dominoes. They all have names. And like I said, this is what I made my students memorize, and I still feel badly. They have like protein B, and protein B comes in, and when it releases an enzyme, it activates C1, which releases an enzyme that activates C4, and then that enzyme activates C2. Do you see how it gets confusing? Oh, it's horrible. It's like alphabet soup. The enzymes then are basically signaling other proteins to help in, and all of these little dominoes keep falling because they've been triggered. Now, there's three main ways that complement can be activated. And again, these three ways can be the classical pathway, the mannose pathway, and the alternate pathway. The classical pathway, this is what I just mentioned, that's where an antigen comes in. And again, an antigen is any foreign substance or a foreign cell comes in. Or I can have bacteria come in that are using or releasing mannose, that's a type of sugar. So we have, for example, staph. Staph loves to eat mannose, right? Remember our plate, mannitol salt, mannose, okay? So mannose is an example of a staph infection that would trigger a complement. And then also the alternative pathway, which can be triggered where the pathogen has some sort of protein on its surface. So not the cell itself, but a protein on its surface that triggers. We use these, for example, for vaccinations. Think about COVID. Our vaccination for COVID isn't based on the entire virus. What's it based on? The proteins and the spikes, right? So we're targeting the corona, the spikes. So that would be the alternate pathway. You don't have to memorize all of these steps. I have become nicer in my old age, okay? Like if you were here before, you would have, 15 years ago, you would have had to memorize this. Instead, I just want you to know that it's like a sequence of dominoes. Okay, so let's check your understanding. Now I have different options on here. You guys just have the questions on yours. I have the different options just to kind of streamline our thought process. What purpose does inflammation serve? So some of the options I put on here: causes redness, limits pathogen progression, increases healing, or all of the above. What do y'all think? All the above. Excellent. And if you want to get this slide in all its glory, you can take a picture of it if you want. Why does a chill indicate that a fever is about to occur? The body temperature has dropped. The hypothalamus has reset to a higher temperature. We need to chill a little more in life, especially on exam days. Or pathogen slow. Yes, yeah, like that. Or pathogen slow blood flow resulting in colder body temps. Which one of these is correct?
B.
So the correct answer on this would actually be B. The reason we get cold, remember, our hypothalamus is like our thermostat. It's been moved up. And now everything moves with it. So with a fever, it's not like my body temperature has changed. I'm at 98.6. It's still 98.6. But the thermostat changes, and that tells my body, hey, 98.6 isn't enough. Let's up it. So I haven't gotten colder. If anything, I'll start to get warmer now. Pathogens, they don't slow our blood. If they did, we'd all be dead. Every time we get sick, we'd be like, oh, it'd be horrible. It'd be it. All right. The signs and symptoms of hay fever occur when pollen leads to basophil activation. So let's see back to our basophils. What are the effects of this activation? Do the basophils release histamine, histidine, histamine, that's for hay allergies, or none of the above? And by the way, C is a joke. There's no such thing as histamine, but yeah, so remember, it's going to be releasing histamine, right? So histamine. Remember, an easy way to remember that is when you buy allergy medication, you're buying antihistamines. So it's an easy way to remember that, okay? So this is just kind of a summary, the signs and symptoms of hay fever. The reason that the histamine is there is because, remember, the basophils produce histamine. These are the same questions, I'm just putting it together. The basophils release histamine. That causes inflammation and it triggers an immune reaction. This is why when you have an allergy, your eyes swell, your nose runs. The idea is that your body's trying to flush out whatever is trying to invade, which is the bane of our existence during springtime. Okay, so we got through innate immunity. So let's take a look now at adaptive immunity. Okay, so adaptive immunity is going to be different because it's going to be where we're more involved in our process and it's more specific. And what I mean by that is it's not just a general shield anymore, it's like a set target. So our bodies are going after a specific virus or a specific bacterial cell. And so again, when we talked about innate immunity, the big difference between these two is that one is just something that's not targeted. It affects every pathogen the exact same way. Adaptive immunity is where our defenses are targeted. They're going after a specific host or antigen. They're ignoring everything else in their path. So in other words, when my antibodies for COVID see COVID, They will go right by flu. They'll go right by chickenpox. They don't care about that. They're after COVID. That's their target. So even though other viruses and bacteria may be present, they're not looking at that. They're only looking at their target. So that's the difference between innate and adaptive immunity. Okay, innate will take care of everything along the way. Now, adaptive immunity is something that we actually build over our entire life through our experiences. So different organisms that we're exposed to will trigger our immune system. And so because of this, our adaptive immunity is always changing. We rely on our genetics to help us produce our antibodies. So adaptive immunity is going to be part of that. And the other thing about adaptive immunity is that the first time we get exposed to something, our reaction tends to be very slow. Our body learns from it. It has to get the information and process it. But then the next time we get exposed to that same organism, we have a much faster result. Think of it like this class. The first time I show you something, you're like, okay, I'm not really sure. And then you go home and study it, and you're like, okay, I got it, right? I mean, think about metabolism. The first day of metabolism, not a good feeling. But by the exam, y'all nailed it, okay? So it's that kind of idea, or our bodies do the same. They have to learn, and then they get specific. So because of that, we have memory cells. And these memory cells remember all of the antigens that we've been exposed to, and they remember how to respond to them. And antibodies that work really well, we make extra copies of them. We make clones of them. And then that clone system allows us to get an even faster response. This is what's behind the idea of developing vaccines. When we create an antibody that works against COVID, we create a whole bunch of copies of it. And then over time, if we go without being exposed, that number starts to drop. Our body starts to make room for new antibodies. This is why we have to get a booster shot. We have to remind our bodies, oh, no, we've got to stay vigilant. Here's that antigen again, and our body's like, okay, and it makes more clones. Does that make sense? Our body's very efficient. It doesn't want to have things it doesn't use. This is also why we need booster shots for things like getting shingles. Our body has all these antibodies against chickenpox, and they tend to last pretty long. But when we're around our 50s, our bodies goes, do I really need this anymore? I'll dump these antibodies. And it'll get rid of some of them, and then that's when shingles can come back. So that's why we get booster shots at certain points of our life. Okay? In case you're ever wondering, like, well, why do we get boosters? Sometimes every year, and then sometimes it's like, every 10 years, or sometimes it's every 50 years. It depends on how long those antibodies last in our body. And some naturally last longer than others. So in this chapter, our main cells that we're going to be focusing on, Brianna.
What determines if they last longer?
Genetics. It's all genetics, and it's about the size and the build of the antibody. And the idea behind that is that we don't want to have antibodies present that are taking up a lot of room. And so our bodies tend to kind of recycle them. But if we have an antibody that's kind of smaller, it's not taking up as much room, it'll last longer. So, and it's all genetics. Some people, for example, like with COVID, some people are getting boosters every 6 months for now. Some people were getting boosters every 3 months because they found that their immune system wasn't stabilizing. And so, again, it all just depends on what we have inside of us. Great question. So our B and T cells are really going to be the stars of our adaptive immunity. They're going to work together. And the B cells are what we call humoral immunity. This means that they're antibodies that are circulating in the blood, that are going to be our memory cells, and they're going to be extremely specific. Our T cells are what we call cell-mediated immunity. The reason for this is T cells, they're not necessarily circulating looking for specific antigens like the B cells do. So B cells will circulate looking for flu. B cells will circulate looking for COVID. T cells will circulate looking for a problem, okay? These guys are cell-mediated, and what that means is they respond to other cell responses. So if a cell gets infected with a virus, it'll send out a distress signal. The T cells are the ones that detect that distress signal, zoom over to that cell, and they'll destroy that cell, or they'll mark it for destruction. These are also the ones that will interact with complement. If complement is attaching to a cell, the T cells will zoom in and help finish it off. So they respond to our other cells. Does that make sense? So B cells respond on their own, T cells respond to our other cells. Now they also work hand in hand though. When T cells find something to target, so other cells are saying, I'm infected with a virus, the T cell finds that virus, it'll get the virus and it'll actually show it to the B cells. It presents its proteins on its surface. And it's also used in as kind of a teacher cell. You can think of T for like teaching as well, because it'll show the B cells what to attack. So we'll talk about this. They work together. So T cells are pretty awesome. Does this make sense then why HIV is so devastating? They're taking out kind of our first responders. They're taking out our cell responders. So they're wiping out just a huge part of our immune system when we have HIV. So as far as the formation of these cells, well, they're both going to start as a little stem cell, and then they're going to differentiate. And when they differentiate, they're going to move to different locations. So what's going to happen then is they're going to move from the bone marrow, if they're T cells, to the thymus. And once they're in the thymus, that's why we call them T cells, T for thymus, they're going to develop in the thymus, and then they'll get released as a mature T cell. The B cells, think of B for bone marrow, they stay in the bone marrow as they start to mature. And then they're going to do a process called differentiation. And what that means is they're going to become different types of antibodies depending on what we need them to be. They're going to respond to what our current body situation is. Then our B cell gets released, and it will start to also circulate with T cells in our lymph system, in the lymph nodes, and in the spleen. So we're going to have this little circulation process. So our B cells then are going to be the ones involved with antibody production. So B cells are going to be involved with antibodies. T cells are going to be ones that are going to really target cells specifically, and they're going to target non-self cells. So in other words, viruses, bacteria, anything that is not your own cell is going to get targeted by a T cell. Okay? B cells are antibodies, T cells are targeting. Now, with our B cells then, as I mentioned, they grow in the bone marrow, so think of B and B cells in bone marrow. They then are going to mature to become true antibodies, and they're going to respond to an antigen. Remember, an antigen is a foreign substance or body that causes an immune response. Okay? Now with this then, let me ask you this. Which cells are produced in the bone marrow? B cells, T cells, or both B and T cells? B cells. Yeah, our B cells are going to be the ones that are going to mature in the bone marrow. Okay. All right, let's talk a little bit more about our antibodies. So we're going to talk a little bit about our B cells first. Our B cells are going to respond to antigens. And again, an antigen is anything that causes an immune response. So an antigen can be a pollen, it can be a bacterium, it can be a virus. The antigens that really trigger our immune system the most are ones that are foreign proteins. So if we have an antigen with a strong protein marker, we're going to get a strong immune response. Also, sugars, polysaccharides can trigger an immune response. Now the reason these are important, remember when we talked about our cells and I told you we had our plasma membranes around our cells and we had little surface proteins and they would be able to recognize themselves as self when they slid past each other. I told you they did like a little secret handshake, the glycocalyxes and things like that. Does that bring back memories? That's what these guys are reacting to. There are different proteins on the surface. There's different sugars on the surface of these invaders. And so they're not recognizable as self. And so that's why they are getting targeted. They use these proteins and sugars as a mark. Yes?
Like a capsule?
It could be a capsule, it could be a cell wall, it could be another plasma membrane. We could have other animal cells get inside and they would still attack it, but it's just not self. Anything that's not self. Okay? So antigens can get inside of our bodies then from the environment. So we can breathe them in, we can eat them, we can drink them. They somehow get inside of us. They can get through a cut. And they can stimulate our immune response because, again, they're foreign. They're different from one of us. So for example, if I have a cat allergy, if I'm petting a cat, I can inhale some of the protein cat hairs. I can get some on my hands and then touch my eyes. These are going to introduce proteins that are foreign into the body. Now, for some people, those antigens that I just introduced, our body will destroy them, but it doesn't elicit a bigger response. It recognizes that it was an irritant that was destroyed, but it wasn't an attacker. Does that make sense? It was something that was neutralized, but it wasn't an attack. For people that have cat allergies, though, which I used to be one of them, I now have cats, but I used to be one of them. If I touched a cat, it was like poof, like my face would swell, my eyes would swell, I couldn't see. So for my body decided it was an attacker. So not only did I get a neutralizing effect, I also got a whole bunch of basophils involved, and I got histamines being released, and an allergy response. So they decided it was something trying to kill me. You know, they're trying to protect me. So how do you get rid of allergies?
You keep exposing yourself.
You keep getting exposed. The idea is we teach the body that this is not attacking you. This is the same thing that happens to people with peanut allergies. When they consume peanuts, the protein in the peanut, for whatever reason, their body decides is something foreign and attacking. And they elicit this huge immune response. And it can be deadly in some cases. The response is so strong it can kill them. We call that anaphylactic shock. Okay, their body responds over the top. So one of the ways we can cure peanut allergies now is to expose people to peanuts in a controlled setting. And then that builds their immunity. It's like their body reacts, but then it realizes, okay, nothing bad happened. And then it reacts and nothing bad happened. And after a while, the body just gets tired and it kind of learns to ignore it. You had your hand up, that's fine.
Oh, you answered the question.
Oh, okay, was it about the peanuts and things?
Yeah.
Yeah, go for it, Jen.
So I know this girl, and she's like literally allergic to like everything, like food wise. So she can only eat really like rice and like meat with no seasonings, and like, but before she like found out, and they just like cut her off of everything because she was getting the migraines. But don't you think they should have just like weaned her so that like eventually she wouldn't be allergic to every single food item?
Right, so probably what they did is they stopped everything just to be safe, to let it clear out of her body. And maybe what they are going to do, and this is what they usually plan, is they would start to reintroduce food back slowly. So they'll reintroduce a group and they'll see how she does. And then they'll reintroduce another group and see how she does. And they'll keep doing slow reintroduction, but they have to have a period of time where it has to be cleansed.
Okay.
And then they can reintroduce. And then they'll watch and see which ones are causing a reaction. And then they'll narrow it down. So that's usually what their plan is. Yeah.
So with people who are like deadly allergic to food, can they still reintroduce the food or not just?
That's a great question. It really depends on their bodies. So for example, I have 2 nephews that are allergic to peanuts. Deadly allergy. To the point where one time I got them a soda out of a vending machine. I didn't even think about it. It was a soda machine. Beside it was a peanut and snack machine. Well, the same person loaded both machines. I got them a soda out of the soda machine, and there was apparently enough peanut residue on the can that they both started to go into anaphylactic shock on me. They were like 8 and 10 at the time. Luckily, I had their EpiPens in my purse, but still, I mean, it's something where it's like, how does that even happen? It's that little bit. Okay, so fast forward, they're now in high school, and they both started peanut aversions. And so what that means is they go to the doctor, they get they start off with like 1/24th of a peanut. 1/24th. It's like a speck, and you swallow that, and then they wait, and they're monitored to see if they have anaphylactic shock. And then if they are, they administer and go down a little bit farther. They would go to like 1/40th, okay? One of my nephews responded great. He now can eat peanut butter. As a matter of fact, he hates it. He has to take a tablespoon of peanut butter daily to maintain his tolerance. So he was like, I didn't know I'd have to eat peanut butter for the rest of my life every day, but he has to. If he doesn't, they're worried that it would come back. His brother, they never could get him past 1/24th. He would go into anaphylactic shock every time. So he's just going to have to live his life with aversion. We're going to have to make sure that he never gets around it. And again, they're brothers, but their genetics were different enough to where one of them could get through it and one couldn't. Yes?
So certain allergies can be passed down from genetics.
So it's not necessarily passing down, it's your body, the whole allergy thing is how your body reacts to something. And our genetics tell our bodies how to react. So some people are going to have genes that tell them to react more, and other people are going to have genes that don't. And for whatever reason, the boys have different genetics. Yeah.
Could that be the same with people who say they have like a gluten intolerance or allergy because they consume gluten very slowly?
Yes. Yes, exactly. And that's what happens a lot of times with gluten allergies is it's something that builds up over time. And so a lot of times people didn't even know they had a gluten intolerance until they became an adult and they're having digestive issues. And then when they have kids, they'll limit their children's gluten until they can get them tested as an adult, just to be safe, because they don't know if they got it or not. Yeah. Okay, so all of these things then are antigens that can trigger our responses. So we can use these as learning tools. So for example, we can introduce antigens to the body, and that is the process with vaccinations. We give you a controlled amount of an irritating product, a virus, a bacteria, and we train your immune system to respond. So your immune system learns to respond. So that's what we're trying to do when we do vaccinations. Again, whether or not somebody reacts to something, it just depends on their genes. This is why some people are allergic to shellfish. Some people are allergic to peanuts. Same idea. Their bodies are recognizing that as foreign. Haley.
So my cousin has really, really bad allergies. Like she's allergic to like temperature changing, like with water.
Mm-hmm.
And she's worried about like getting the vaccine. So like what would be like a smart, like safe way of getting it without like the
Has she
fear of
had
allergy?
issues with any of her previous vaccines?
I don't think she has.
So if she hasn't had issues with her previous vaccines, she shouldn't be worried about this one.
Okay.
Because that's one of the first things they'll ask you is, have you had issues with previous ones? And then they're going to ask you, what were the issues? But if her immune system is one that can learn and be trained from a vaccine, she's fine. Oh, it's a totally different process. But I understand that concern because you're always worried. You're like, okay, well, what's going on?
Yeah.
So our antigens then, when they come into the body, we need to think of the fact that a lot of these things like pollen or cat hair, we can see them with the naked eye, but there's more even there that we can't see. So for example, on that pollen or on that cat hair are little tiny specific binding sites that are called epitopes. These are different types of proteins that our immune system responds to. And I'll show you a picture of what an epitope looks like in just a second. So the epitopes are binding sites for our antibodies. And everything out there has different proteins on the surface. Cat hair, shellfish, everything has these proteins or these epitopes. And our antibodies respond to those. We're then going to make B cells that are specific for those epitopes. And then that's how we build our immune system. And I'll show you a picture of this in a second. I'm just giving you the background and we'll get a picture. We also can have some substances out there that we refer to as haptens. Haptens are ones that if you get exposed to them, they're like a very small protein. And in some people, they don't react at all. But in our bodies, sometimes those haptens, for whatever reason, will bind with complement. And remember, complement is like dominoes. And when they bind with complement and a carrier protein, they trigger an immune response. An example, poison oak. Poison oak is an oil, right? For some people, they get exposed to poison oak, and we refer to poison oak as a hapten. The reason for that is because it's not your typical large protein marker. Instead, it's this little substance that by itself doesn't trigger a response. But if it comes into contact with a certain molecule in our body or with a complement molecule, in some people, it will trigger. And then what happens with poison oak? If you're allergic to it, what happens? Yeah, you get these rash, you get itching, you get upset. Everything's like inflamed. Your immune system's reacting. Some people, like myself, I'm completely immune to it. I can go sit and roll around in poison oak, and it is no big deal at all. Actually, my friends hate hiking with me because I don't even watch for it. I just walk through it, and then they're like, why did I hike with you again? It's like, I'm sorry. So for me, I get exposed to these haptens, but I don't have the carrier proteins to make them bond. So they don't respond to it. So let's look at a picture of what this looks like so it'll make more sense. Okay, here's a picture of a bacterial cell. This is not true to size. Okay, they're not this big. Okay, on this bacterial cell, it has a whole bunch of protein markers. Okay, and they can have even more than this. This is just an example. Now, these protein markers are going to be all over the cell, and that's what we're going to refer to as our epitopes. Epitopes are the proteins. Now check this out. Here's our antibodies. And notice we have different antibody shapes. Do you see that? So I have antibody B here, which has kind of this semicircle shape. So it's going to be able to attach to any proteins that are semicircle. Antibody A has a triangle shape, so it's going to attach to any epitopes or proteins that are triangular shaped. So in other words, different proteins will trigger different antibodies. And you can have multiple antibodies attacking the same target. Okay? This is where we get a faster immune response. This is what happens with COVID then. When we build our antibodies, we build several types to the spike proteins. This is why when you get sick, you recover faster if you've had the vaccine because you're attacking it as soon as it shows up. Okay? For people who aren't vaccinated, they have to learn. Their antibodies have to learn what these proteins are. They have to find the right proteins that have the right shapes, and it takes a learning process. Okay? So those are epitopes. Haptens are a little different. Haptens are kind of like, think of like an oil marker, or think of something that has fewer proteins. It's very small. If it comes into our body, so this is the example I gave you was poison oak. If it comes into our body, and if we have a particular carrier marker or molecule that binds with that hapten, it will create a substance that will trigger complement. So if my hapten comes in by itself and I don't have a cell for it to bind with, I won't react. So for example, people who are immune to poison oak, they get exposed to haptens, they genetically don't have this carrier molecule to interact, and so they never get to here, which triggers complement. Okay? If I do have this carrier molecule, and the carrier molecules vary from person to person, that's why I'm just being general, then I will get a reaction. This is the same thing that can happen with shellfish. Shellfish have a hapten molecule. When I consume clams or mussels, I get these protein haptens. For some people, they have molecules that will bind with those protein haptens and create something that triggers an immune response. Okay, other people don't. That's why we have some people with shellfish allergies and other people that don't. Okay, so our antibodies then are going to be specific because they have specific shapes. And they can only bind to specific epitopes on the antigens. So each B cell out there is going to be specific for a protein marker on an antigen. And hopefully that picture kind of showed that. They can be triangular shaped, semicircle, but they're specific. We encounter, on an estimate, about 1 million different types of antigens in our lifetime. That's huge, probably way more than that. And that means we have to have antibodies for all of them. Can you imagine that? So we have millions of antibodies. Now the antibodies then, the way that they're formed is they can be different for what reaction or what area they're supposed to circulate. There's a lot of different sizes, shapes, concentrations, and this is why I mentioned that the body will get rid of some sooner, keep some longer. It just depends on how much storage room the body has. They are all made of the same protein, which is called Ig or immunoglobulin. That's specific for an antibody. It all has the same component that builds. And they're all going to have an area referred to as the constant region, and the constant region is going to be the same in each type of antibody. We're going to learn that there's 5 types of antibodies. So if I go from one antibody to a different antibody, they'll differ, but in that grouping that they're in, they're all the same. I'll explain. So for example, we have antibodies that are called the IgGs. All of the IgGs are the same with their constant regions. All of the IgEs are the same with their constant regions. They differ from one another, but they're the same as their group. Does that make sense? Okay, so if I have group A and group B, they're going to be different from one another. Okay. Now, they all will have kind of this Y shape to them. The Y shape might be in a different type of arrangement depending on which antibody we're looking at. They're going to have what are called chains on them. Chains are basically sequences of proteins. Remember when we talked about DNA making proteins? The chains that are present are sequences of proteins that let the antibody know what to target, what shape they're going to be, and what their target is going to be. Okay, so these chains are proteins that will interact with epitopes. And we're getting a lot of terminology, so I want to make sure we're still okay. The chains are going to help the antibodies know what to attach to. Okay, so just to make sure. All right, so on each side, they're going to have identical light chains, and then they're going to have identical heavy chains. These chains then will make them specific for their targets. Now, as I mentioned, there's different classes or groups of antibodies. They all have immunoglobulin still, and we designate immunoglobulin as IG. So IG is immunoglobulin. Where the classes vary is depending on how they circulate, how they look, and what they target. So we're going to talk about each of these classes. So each class is going to have their own special projects or special targets. So let's start with the first one, the IgG antibodies. IgGs are going to be the most common ones in our bodies. They make up about 80% of our serum antibodies. So if I did a blood test on you and looked for antibodies, about 80% of those are IgGs. IgGs look like a simple Y shape. So they're singular, they're little mono antibodies, they're a single Y. And that's why they're called a monomer, single Y. They're going to circulate in the blood. They can cross the placenta. So these are ones that are found in breast milk. They're found in placenta. They neutralize microbes and toxins. They help target antigens for phagocytosis. So they act as little flags. They'll attach to a cell, kind of like what we see here, and then the T cells will come in and help target. They're going to help work with complement. They do what's called opsonization, and what that means is that it's a specific target against specific types of viruses or bacteria, so they help streamline. Think of it as like streamlining attacks. They protect our newborns. When our babies are born, the antibodies that the mother has crossed the placenta and it's now in the babies, so they protect the newborns. They offer this huge increase anytime we see an active infection, their numbers go up. So if I do a blood test and I see a high number of IgGs, that tells me that you're currently ill, that you're fighting something at that time. And what's interesting is I can look at your IgGs and they can tell me your history. I can see what you've been exposed to in the past based on your response to different types of antigens on a blood test. Now, go ahead.
Does that mean that these IgG antibodies are not found in the placenta after birth?
They're in the placenta, but they've crossed through the placenta and they've gone into the newborn.
So when mothers choose to do the whole placenta pill thing, does it have benefits to it?
To be honest, not really. It's kind of a psychological thing more than anything. It's a placebo effect.
Right.
And the reason we say that is when we've looked at the placenta levels. So, for example, when we check people's blood levels, we don't see an increase when they're taking the placenta. So what she's talking about is there's a lot of women that'll dry the placentas, they'll turn them into capsules, and then they'll take them, thinking that it's going to replenish iron, replenish antibodies that they gave to the baby. But we don't see that. Your body is set to have this child and to expel it, and we don't need any other nutrients. We're good at that point, at least from our current findings. Yeah, great question though. Okay, so anyway, they can also show us past exposures, which means that this is why if you get a COVID test and you're positive, they tell you not to retest. If I get a COVID test and I see IgGs, that tells me I have an active infection right now. Okay, my IgG level is going to be high, and it's going to stay high on average for about 3 to 4 months after COVID exposure. So what this means is if I have a positive test, I run the risk of that test being positive falsely for like another 90 days. So a lot of times people will tell you, oh, get another test and see if you're clear. That doesn't always work because it'll show that we still have a high level of IgGs as our response. So they're not, that CDC says, no, don't retest. Yeah. What's that? Oh, thank you. Thanks for letting me know. I don't think I have anybody in here. I had a person popped in and then they popped back out. So I'm like, okay. All right, we're just going to go. Thank you. Okay. Also, we have IgMs. IgMs are the next set of antibodies, And these are referred to as pentamers because there's 5 of them, 5 little Y's arranged. They're found in about up to 10% of our serum. And these are secreted initially during a primary infection. So my IgGs are there when I get sick, and they stay long term, so several months. IgMs are there when I get sick initially, and their numbers will increase. That also show that I have a current infection. Now, IgMs cannot cross the placenta. We don't know if it's because of the size that they are or what, but they cannot go into the placenta. So they're not going to be associated with newborn protection. Now, this is important because if we have a baby who is sick, we don't want to use IgMs as a detection in the respect of thinking that, oh, this was the mom's IgMs. We would want to make sure that we understand that if they're sick and they have IgMs, that means that they're really fighting something. They're on their own. It's not something they inherited. Does that make sense? So if a child's sick and I check them for IgGs, those IgGs could be from the mom. If I check them for IgMs, though, that's all them, and that means that they really are ill at this time. And I need to do something right away, an intervention. Okay? So you use this as a marker of a current infection. IgAs. IgAs are at about 15% of our serum, and they're found primarily in our mucus. A lot of times they make what's called a J chain. It's like 2 that are hooked together, so it kind of looks like an inverted J. They tend to be seen in mucus. They're also found in breast milk excretion, so another one, they're like the IgGs. They can be found in our tears or in our blood, so they help with getting rid of toxins and microbes and targeting them for destruction. So if I was to check, for example, your mucus, I should see a lot of IgAs that are present. Breast milk, IgAs are also present. IgDs. IgDs are ones that we're still trying to learn about. They seem to play some sort of activation role with our B cells, meaning that when B cells are first learning what to attack, we find a lot of IgDs around them, but we're still not sure of what their exact function is. We're still trying to learn. They're only at about a 0.2% in our serum, so they're not found in a high amount of our serum. And we think, again, they're somehow involved as like an instructional assistant. They're helping our B cells learn more so than targeting other cells. They're more so than targeting like foreign objects. We're still trying to learn what they're doing though. And then IgEs. IgEs are the ones that are associated with parasitic infections. So if I do a check and I see a high amount of IgEs, that could indicate parasites. They're ones that are associated with allergic responses, like anaphylactic shock. These are the ones that will all of a sudden increase if I'm having a reaction to peanuts or shellfish. These things will shoot up. So if I see an elevated IgE, that means that my patient is having some sort of internal response to something. And usually it's something that could cause shock, or it was something that was very aggravating to their immune system. So as we learn about what these antibodies do, we can start to use them to give us information. So for example, IgMs. When we first get exposed to any kind of infectious product. Did you guys need me to go back? Did I go too fast? Sorry. I'll go back one. You guys are okay with that one? Okay. So when we first get exposed to any infectious particle, kind of like we said, our neutrophils were our first responders, remember that? Our IgMs are the first antibodies that we see a response with. IgMs will spike up when we have a current infection with something. Now the IgMs will trigger additional antibody production. So as soon as the IgMs shoot up, the next antibody that shows up and starts increasing in number are the IgGs. The IgGs come in and they help get rid of whatever was triggering our immune response. Then the IgGs hang around as memory cells. They're persistent. The IgGs then are the ones that we don't want to use for COVID testing, for example, because they're going to be present for 90 days or 120 days. Again, this is why we tell you not to double test if you're positive. IgMs are better antigens to use to see if you have a current infection. Meaning that if I see a high number of IgMs, that means I'm fighting something right at that moment. If I see an increase of IgGs, that means I have fought something. I might still be fighting it. I might actually have cleared it, and my IgGs are still high because they're still like on high alert. It's kind of like if you have something scare you, your adrenaline goes up and you still kind of feel that feeling for a little bit. Like if somebody almost hits you in your car, if you ever had that happen, you're like, oh, and then you just like have that adrenaline rush for a little. That's the IgGs. They're like that after adrenaline rush. Okay, the IgM is like the initial one. Okay, so you can think about it that way. So let's put this together then. I have an infant who is sick. The infant has a high level of IgGs. Could that infant have inherited those from the mother? Yes. So I would want to look at the mom and see if she was sick as well, because that newborn or that infant, those IgGs could be its mother's as well as its own. So that's not going to give you as much information. But if I look at this infant and I run an IgM test, does that give you more information if it's sick?
Yes.
Yeah, because that's going to tell me that it's fighting an infection on its own. How do I know? IgMs can't pass through the placenta. So this child is fighting an infection on its own. It's producing antibodies. Okay. Now let's say I'm going to do a COVID test and I want to know if you have COVID right this second. Which antigen should I target, or which antibody should I target? IgM, exactly. That's my primary response. Excellent. If I want to know if you've ever been exposed to COVID, which one could I use? IgG. Those are more of my secondaries. Think of IgGs as kind of like footprints. If you walk in the sand, you leave footprints behind. The IgGs will remain elevated, meaning they're visible footprints for a while, and I can see what you've been exposed to, like during the last year. Isn't that cool? I can check your IgGs and be like, oh, did you have the flu? Oh, did you have COVID? Oh, did you have this? I can check. These are like our little memories. IgMs tell me what you've been exposed to right now. They're like in the moment. Yes?
Are these different markers and like for people when they would get tested for the wrong one?
Oh, for the breast cancer and
Yes.
stuff? So again, those are self-markers, so it's a little bit different.
Okay.
Yeah, so those are looking for specific proteins on our own cell surfaces. So not necessarily antibodies, because we're not targeting the cancer. So it's a different marker system. Good idea. I like how you're kind of putting stuff together though. Okay, so the role then of our antibodies is that they are going to help make sure that they neutralize any threats. So our antibodies can grab toxins and remove them from the blood. They can grab bacteria and viruses and destroy them. They can cluster them together and destroy them. They can try to rip cells apart. So they're trying to neutralize threats. Where we run into trouble is sometimes our immune system, when it's responding, can jeopardize our lives though. So for example, one of the things we have to be careful of is blood typing. Why is blood typing so important? Well, it turns out that on the surface of all of our blood cells, we have specific proteins. That makes us either type A or type B. What about type O? Does O have proteins on its surface? No. Type O is an oddball. It doesn't have any proteins. Okay. So let's say that I'm a type B person. So all of my blood cells have B proteins. And I get a blood transfusion from a type A person. So now they have a different set of proteins. So I have blood cells coming in with epitopes that don't match. Is that going to trigger an immune response? Oh yeah. My antibodies are going to go and grab the epitopes on those blood cells. They're going to help with opsonization. That means they're going to grab and cluster and group. So what do we start to see? We see blood clots. And blood clots form all throughout the body. Everywhere there was a blood cell that traveled, they're going to start clotting. And it's like those domino effects, like we talked about earlier with our complement. Those clots get bigger and bigger, and they're going to stop your blood flow. They're going to cause organ destruction. They're going to cause death. That's why it's so important to do protein testing with blood typing. Okay? Yes, go ahead,
Is
Laura.
there a way to reverse that? Or like what's happened to them before?
There's not really, you can try to put in immediately the right type of blood and try to dilute it out. That's your best bet, is just to try to dilute as fast as possible and remove as much blood as you can and filter in new blood, but you're still going to have some damage. So depending on what your medical access is, you can probably stop death, but you're still going to have some tissue damage, definitely. And depending on where that blood traveled, if it went to the brain or whatever, it can be more devastating or not. But good question. But you tried to, basically you're just trying to dilute what you just did as fast as possible. Okay, so with this then, our antibodies are involved in grabbing lysine, trying to destroy things. Now, this active immunity then is going to be where our antibodies are learning what to attack. And we do this our entire lives. And active immunity is where we form antibodies from exposure, and this creates long-term memory cells. Meaning that if I'm exposed to something, my body will remember it and it will attack it again. So, an example of this would be, for example, if I get the flu. If I get the flu one year, my body will attack that virus type. I will have antibodies in place, and then I might not get the flu again, that particular strain, for like 5 more years. This is why a lot of times when we get the flu, we're usually okay for a couple years. Where we run into problems with the flu is there's multiple strains. So if I got one strain, I have protection against that strain, but if I get exposed to a different strain, I can get the flu all over again. Okay, so there's different strains of flu. Fifth disease. Fifth disease, for example, once you get it, you build up antibodies towards it, and then the next time you get exposed, you're not going to get it again because you have memory cells in place. Chickenpox. If I had an active case of chickenpox, meaning I got it from somebody, naturally in the wild, I would build up these memory cells, and the next time I see it, I don't get it again. This is why you often hear, oh, you only get it once in your life, right? It's because I have these memory cells in place. Now that's active immunity. That means I'm triggering a response and my immune system is creating antibodies. Passive immunity is a little different. Passive immunity is where I'm giving somebody antibodies. So they're not producing them on their own. Instead, I'm giving them to them. So an example would be something like breast milk. When you're breastfeeding your child, your antibodies are passing to this infant or child, however, whatever age you want to stop. I know some people breastfeed until their kids are like 5 or 6. Whatever, that's fine. Whatever you want to do, that's fine. No judgment. But the idea is that your antibodies are passing. So I was at the, my thing is if they can ask for it, it's too old. That was just me though. I was like, no. But that was just me. But again, no judgment there. So some people, as a matter of fact, right now with COVID, there's a lot of women, there's a whole movement of women that are breastfeeding all of their kids. They're expressing breast milk for all of their kids to try to give them natural antibodies. So again, it's like whatever you need to do to feel safe, that's fine. So milk contains antibodies. Now when I pass this to the infant, they're not doing anything. They're just getting antibodies given to them. Because of that, they're short-lived. Because they didn't make them, they're going to be in their bloodstream, they're circulating, but their body's going to destroy them after a while because it's going to be like, what is this? I didn't make this, and they're going to get rid of it. Okay, so it's something that is a short-lived process. Now, I can take active and passive immunity, and I can break them down a little bit more. So active immunity, remember, I'm producing my own antibodies. We can get active immunity then through natural exposure. I'm riding the bus, somebody sneezes at me, boom, I get exposed to a cold or a flu. I build up antibodies for that strain. That would be natural. Or I can build up antibodies artificially. I want to protect myself against COVID. I go and I get a COVID shot. The shot is exposing me to a carefully regulated amount of viral particles, so I won't get sick, but I will get a reaction or a response, and I will build antibodies. Okay, so that's artificially, meaning that I was given it under a controlled setting, as opposed to somebody sneezing in my face. Okay. Passive immunity, it can be naturally acquired through things like breast milk. Okay, so this baby's getting antibodies given to it. Or we do the same process when somebody is bitten by a rattlesnake or stung by a bee and they're having a reaction. we can give them antibodies from organisms that have already been exposed to rattlesnake venom or that have already been exposed to bee venom. And those antibodies then can be used to help break down the toxin or venom that they've been exposed to. Right.
Like an EpiPen?
Yes, exactly. Same idea. Yep, same idea. EpiPens, they don't necessarily have antibodies in them. EpiPens basically just tell your immune system to stop. It's like shutting it off. It's almost like an antihistamine, but it's like on steroids. It's telling your immune system knock it off. It's basically like a huge steroid. Any questions on this? So, for example, when there's an Ebola outbreak, they have found that using artificially acquired passive immunity actually can help with recovery. So they will take antibodies from people who have recovered from Ebola before, and they will give them to people who have Ebola at that moment. And it seems to help their bodies break the Ebola down faster and to stimulate antibody production. So again, it's a short-term effect, but it can help maybe keep them alive enough while their body is doing active immunity, creating their own antibodies. Yes?
I always think around this weekend, when people donate blood, does any of that stuff like good?
Great question. So she said, well, what about people that donate blood? So when people donate blood, one of the things that they do screen for is they do check to see what kind of antibodies and antigens they have. But remember, when you do a blood donation, it's not usually used right away. It's stored. And so any antibodies or antigens that were in there usually break down pretty fast. So you're not going to inherit what they had, unfortunately. I wish you would, right? You could have like Superman blood, which would be awesome. But usually by the time you get the blood, it's been sitting around long enough where the antibodies they're not there anymore. They're broken down. So again, this is just kind of a map showing the exact same information. So I can have our naturally acquired, meaning that I'm out there getting exposed or sick, or I have a natural process like breastfeeding, or I can have artificially acquired where it's active, meaning I'm getting exposed to a vaccination, or it's passive, meaning I'm getting something like a snake venom antitoxin. All right. Do you guys, let's see, we'll do vaccination and then I'll go ahead and stop a little early because I know you want to review for the exam. Okay, vaccination. So again, the idea behind vaccinations is we're stimulating the body to create antibodies. We are exposing you to a known viral particle or bacterial cell, something that is going to stimulate an immune response. We do this in a controlled manner, meaning that it's not enough to actually cause a disease. So, for example, think about tetanus shots. Are tetanus shots comfortable? No, they're miserable, right? I mean, let's be honest. You get a tetanus shot and for 3 days your arm's like this, right? So can you imagine how bad that bacteria would be if that wasn't a controlled amount? So what's happening with tetanus? Remember we said tetanus is where you're getting an artificial clenching of the muscles that never relax. So when they inject you with just a little tiny bit, it causes inflammation in the muscle and it causes some of our muscle fiber to contract, which is why our arm feels so darn sore. Now, that's a controlled setting. That's like a small amount. That's a little bit of the toxin being presented there. But because of that little bit, we build these huge antibodies, and then the next time we get exposed to it out in the wild, our bodies recognize it and kill it before it can cause an infection. But I always think about that. If your arm hurts that much from that little tiny bit, can you imagine how bad true tetanus feels? And I told you we had an issue where they had tetanus in Texas. There was a young boy that was not vaccinated and getting tetanus. He was in the hospital for over 9 months. Those were miserable months for him. And his parents were like, oh no, he's good. This is why we don't vaccinate. It's good that he's here and that he recovered. 9 months. It's like, oh man, it's worth getting that shot. You know, I don't want to be in a hospital for 9 months. I don't know about you guys. So we produce memory cells then that are long-term. So things that are long-term that are going to protect us. Okay, so is vaccination an example of innate or adaptive immunity? Adaptive. You guys are saying you're all kind of like, maybe adaptive? I think it's adaptive. Yeah, adaptive immunity. It's something that's a learned response. It's specific. How would snakebite antivenom fall in these categories? Is it naturally active, naturally passive, artificially active, or artificially passive? Excellent. It should be artificially passive. The second B. It should have been a D. Yes. Don't judge me. Don't judge me. Exactly. So artificially passive. Okay, our next slide is going to be B lymphocytes. We will pick that up next time because I know you guys want time to review that. We'll be back here at 10:30 for our lab exam.